Okay, we've got Triana Amen, who's going to be giving a talk on CRISPR-Cas9 and screening for levels of ABCD transporters. And Triana is an assistant professor in advanced cell biology at the University of Southampton in the UK. Her lab studies the principles governing the biogenesis of organelles and how changes in their architecture are brought about by stress and disease biology. So, thank you very much.
Hello, everyone. It's really a great pleasure to be here. Thank you so much for this opportunity to present our research to you today. So I'm an assistant professor in cell biology. I teach cell biology to students, and usually when I teach cell biology, I remind students, and probably mostly myself, that all this amazing complexity, these astonishing structures that we see in the cells, comes from very basic and very few building blocks. And fatty acids is a class, a lipid class, one of these building blocks. And so fatty acids comes in different forms. So you heard from many of the lectures here today. So if fatty acids are medium chain or long chained, so sort of usual chain of fatty acids, then they are amazing building blocks for cellular membranes. They can be used as energy, they can be stored. But if fatty acids are too long, so longer than usual, or a scientific term for that is very long chain fatty acids, then cells do not have many use for them. They're poorly soluble, they're unwieldy as components of many reactions, and they can be toxic when they accumulate, such as in ALD. And so cells have this amazing things that they do with them. So if you look very, very close inside living human cells, what you will notice is that there are these compartments that we call peroxisomes. And these peroxisomal compartments transport fatty acids inside of them, and they have incredible machinery to just degrade them. So basically, cells accumulate these very long chain fatty acids in peroxisomes for their degradations. So today I wanted to divide my talk in three parts. So in one part, I will tell you sort of the learning, the lessons that we learned from studying this transport of very long chain fatty acids inside of the peroxisomal compartments. And then I will tell you how some of the assays that we are developing can be used to study ALD in cellular models, and potentially even classify some of the variants that were discussed here today. So just some very basics. So basically, each one of these peroxisomal compartments that you see inside of a human cell here is a single membrane compartment, and on the membrane of this compartment, you have what we call ABCD transporters. So these are proteins which are really beautiful. They are normally composed of two proteins to form a functional transporter. And this transporter, basically, the job, the function of this protein is to take fatty acids, very long chain fatty acids, from the cytoplasm or outside space and put it inside peroxisomes for degradation, right? So what we learned from initial sort of attempts of to establish some assays, we learned very simple things that I wanted to share with you. So first, if you look at different cell types or different tissues in your body, you will find that different ABCD transporters are present in different copy numbers. So basically, there are some cells where these compartments, peroxisomal compartments, have very few transporters, and there are compartments where you have many, right? And this is basically what you see here. So we can study it using sort of, it's called Western blot, but it's basically we lyse the tissues or lyse the cells, and with antibodies we determine the bulk content of these tissues or the cells. And so the band here, and here is quantification of intensity, represents how much of this protein is there inside your cells or inside your tissue. And so you see, if you see human kidney cells comparing to human bone cells or osteoblasts, you have a significant difference. So there are different levels. And if you look at any other tissues, you will also see this difference. Another way for us to study this is we can look at single peroxisomal compartments and also quantify the level of these transporters on the peroxisomal membrane. So here you have a monolayer of human cells, and the control here is a monolayer of human cells that lack peroxisomes altogether. So they lack the structure where these transporters are, and you can see it in the staining. So each dot here represents a compartment and represents an ABCD transporter on the membrane. And so we can also quantify its intensity on a single peroxisomal compartment. Another thing that we learned from doing these things is that if we look at these levels of transporters on single peroxisomal compartments, and we can do it with another assay that I show you here, we can fuse endogenously using CRISPR-Cas9 a fluorescent protein molecule to a transporter. So here you have it fused to both copies of the functional transporter. And then here you just see that all the endogenous ABCD transporters now are with fluorescent molecules, so we can visualize them in the cells. So whenever we see the green signal, it's coming from a native level of the protein. So what we did here, we looked at the same cell type, but in different conditions. So we exposed cells to hundreds, and here you see 150 different conditions, basically regulating different signaling pathways of the cells. And here's just a conformation in a human fibroblast. I do not think that you will be able to read out these images, but what I want you to appreciate is that the endogenous level of ABCD transporters sort of drastically change depending on conditions, right? So you have conditions where you have significant increase of these transporters on peroxisomal membranes, and you have conditions where you have significant decrease of these transporters. But all of this, looking at levels, does not actually answer the most interesting question, because the most interesting for us was, what is the actual function? So will different levels affect the function of the transport? Because the key component of ALD progression is, first, we have a dysfunctional transport of very long-chain fatty acids inside peroxisomes, and then it leads to their accumulation and subsequent toxicity and effects, right? So in order to establish functional assays, a couple of years ago, we developed probes that you actually see in this movie for live cells. So basically, we developed a probe which mimics very long-chain fatty acids, but we can also see them under a microscope. So basically, it's a fatty acids that are delivered into peroxisomal compartments. That's what you're seeing here. But we can also visualize this process on a confocal microscope, and we can quantify this process as well. So initial probes we called PeroxiSpy. It was done in Swiss Federal Institute of Technology with a brilliant team of chemists, Luc and Milena. And then we have them of different colors. You have the red color or this magenta color. We have a far red one, so for different microscopy applications. And then when we published this initial probes, probably a day after the publication, Professor Stefan Kemp wrote me a letter and introduced me to this wonderful ALD community. And he had a brilliant idea. So he noticed that what we are actually measuring is how substrate goes through the ABCD transporter. And so with a lot of subsequent proof of concept experiment, we can actually say that we are measuring transport, real-time transport in live human cells inside every single peroxisomal compartment, which you can see as dots here, and really, really significantly zoomed in situation here, where you can see it actually crossing the membrane inside peroxisomal compartments. All right, so we formed a collaboration, and together we decided to try to tackle this and try to establish the functional assay. So I will show you some proof of concept experiments that we are working on at the moment. So initially, we used our genetic engineering capabilities to create sort of cell line samples for proof of concept. So we have cell lines that normally expressed ABCD1 or ALD protein. I will use it interchange these names quite a lot. And then we have a knockout, CRISPR-Cas9 knockout of ABCD1 as a control, right? And then we can replace the native copy with stable line with healthy controls, and then later I will show you that we can also replace it with variants of ALD. And we can also do all this in cell lines that already lack other ABCD transporters for non-interference with our assays, because some of them have redundancy in terms of recognition of the substrate, right? So we are doing it in this case in knockout or silencing of ABCD2 and 3. So we want to focus just on ABCD1 or ALD protein. And so here is a proof of concept of this assay. So in this sample, in this cell line, we have only ABCD1 present on peroxisomes, and we are silencing other transporters out. And then we have a control cell line where we have silenced all of them or knocked out all of them, which means that there are no transporters. So this is basically showing you the specificity of our probes, because they will not stain anything without actual transporter being on the peroxisomal membrane, right? And this is quantification here. This basically is a prototype of our functional assay showing that we can measure directly the function of ABCD or ALD protein. And then it becomes more interesting. So if we can replace a normal copy with another healthy copy, which usually makes a complementation experiment for basic cell biology lab, we can also do it with variants of ALD and then measure their function, right? So now we can establish function of the different variants. So I will just, because I will be showing you a lot of structures in the next slides, I want to briefly remind everyone what I'm actually talking about. So basically, in every cell in our body, so we look inside, we have hundreds of compartments that we call peroxisomes. So you see this neuronal cell line, and all the dots here are single compartments. If you zoom in and zoom in again, you will see this peroxisomal compartment, and on the membrane of each of them, you have ABCD transporters of 1, 2, or 3. And then each one of these transporters is this beautiful protein that can be in different conformations. So I chose this open one because it's initial one where fatty acids binds to it, and then it takes some energy, which is these domains for doing this, and then it pushes this fatty acid through inside of the peroxisomal compartment, and then the energy is used to actually change the conformation and push it from the open side to the sort of closed and in, and fatty acid goes in. So that's basically the basic structure of ABCD1 transporter in this case. And now, when Stefan introduced me to this amazing community, I learned of this astonishing number of things that you all guys do. And one of these things is you have the ALD database. So you have all the variants classified in a very thorough and very accessible way to everyone. And so we thought that, well, let's map these variants on this protein structure just to see what's happening with the variants. So here I'm mapping some of the pathogenic and likely pathogenic mutations of variants of ALD protein on the actual structure of ALD. So in red color, every dot here is substitution of amino acids to a different one. And so in orange one, you have likely pathogenic mutations. And then if you superimpose on this the benign and likely benign, and here I'm only showing the point mutation, so I will later show that there are other things like deletions, right? So you can see that no portion of this ALD protein is spared, practically all the domains, including the domains that actually bind fatty acids, the transmembrane portion here, the domain that actually does the energy conversion to change the structure. So everything is covered with the variants, the mutations, right? So we decided for our proof of concept initial experiments, we will take two pathogenic variants. one of them will be in different domains. So one of them in this transmembrane region, and one of them in the energy region of the cell. So we took positions 98 and 514 for that. And so what we did is very simple. So we knocked out the healthy copy of ABCD1, and we replaced it using stable lines with either normal copy as a control, or 98 or 514 variants of ALD. And so even from this blot, so we can measure the levels and then correct our microscopy data by the levels, which is quite important. But even from this blot, you already see that when you create stable lines, they may have so when expression stabilizes over weeks of selections, you can find different clones, but most of the clones will fall into like high expressing or low expressing or medium expressing categories, but some of the variants immediately present themselves as unstable, so we can, from this blot, already actually see which variants are unstable, because you see when you have a variant of ABCD, it can be dysfunctional due to different reasons. So it can be dysfunctional topologically because it just doesn't reach peroxisomal membrane. It can be dysfunctional by reaching peroxisomal membrane but not being able to do the conformational changes. So there are different layers to this dysfunction, right? So here it's interesting because this variant is already not actually reaching the levels of a wild type. But that's I'm sort of going on a tangent here. So basically, we created 3 different cell lines. So one will be our control, where we replaced endogenous copy with another wild-type healthy copy of ALDP, and then we replaced it with different mutations. We know that they are pathogenic, so we know that they are likely absolutely dysfunctional, and so that's exactly what we see in our prototype of our assay. So there is a significant difference. You can probably appreciate it from the images. There is significant difference in terms of staining of peroxisomes. There are a couple of challenges that we are working on. We are trying now machine learning-assisted recognition of peroxisomal compartments, because when they are not stained, how do we know where to find them to quantify intensity profile? So there are a couple of technical challenges that we are still working on, but so far with this, it's pretty clear that we can read out the dysfunction of the ALD protein. So, and then we can even go further. So we can take a couple of steps further and look at the variance of uncertain significance, right? I will not be able to show you the data today, but I wanted to show you that we mapped this variance. And so in white dots here, you can see the variance of uncertain significance sort of superimposed on the all other variants. And again, you see the same situation where no portion is spared. It's equally distributed throughout the whole structure of the protein. And you can also appreciate how many of these variants are actually that would be nice to know if these proteins are functional or not. So I think what we can do with this assay, and what we proposed less than a year ago probably, and we were generously funded by Ella, is to do that, to try to classify this variance of uncertain significance and potentially other variants in terms of function of the ALD protein. So I didn't show you this first portion where we actually do proof of concept experiments of our probes binding as substrate to ABCD1. This is very molecular biology heavy portion, but I showed you a little bit about the models and how we can read out the function of ALDP in the models that we create using CRISPR-Cas9, and with the eventual idea being we can classify many different variants, potentially maybe all of the variants, depending on how long I can make my students sit on the microscope. So we started this grant over the summer, but we already I decided to look at some of the proof of concept experiments for the patient cell. So finally, I would like to address this question whether we can apply this functional assay directly to patient cells instead of creating these models with CRISPR-Cas9. So initially, when we developed the probes, in the first publications, we immediately noticed that probes have diagnostic potential for peroxisomal disorders. So whenever you have peroxisomal dysfunction, which can be a severe peroxisome biogenesis disorder where peroxisomes are just missing from the cells, you immediately see it with the probes because they accumulate in peroxisomes. But even milder cases of PBD where peroxisomes are still present, so the compartment is still there, but there is certain dysfunction in protein import, for some reason we still cannot explain for some of the cases, we can read it out and quantify the differences between the cells. So we decided to do the same and apply it for the ALD. So we took a case with quite an interesting variant here. We have a deletion of a C terminus. So what it means is that the portion of a protein is just missing. And so this portion is outlined here in red color on top diagram. And what actually will be present on peroxisomal membrane is what you see on the bottom. So there will be truncation, so there will be a missing portion of the ALD protein. And we know that this variant is pathogenic. And so initial attempts were to apply our probes to just the patient cell lines without any additional modifications. And we had a significant difference in the import, which you can see here in these images and quantifies kinetics. But it wasn't a difference that it wasn't a very strong difference because we know that this variant is pathogenic. So we saw that the initial attempts were hindered by presence of ABCD2, potentially an ABCD3 transporters. So what we did next is we silenced ABCD2 and 3. So we left only ABCD1 in the cells. And then you can see that if you compare the left and right image in magenta there, you can see that now there is a much bigger difference between the readouts. So what I'm showing you here is sort of our attempts to optimize the assays that we are developing, and with the hope that when we classify the variants, we have maximum dynamic range of our functional assay, right? And then another interesting thing that we found while doing this is I didn't mention, but this incubation with our probes takes only 10 minutes, so it's a very, very fast assay. We just throw them on the microscope on top of the cells, and then we just measure this on the confocal microscope while cells are still alive. So, but if we incubate these probes for a longer time, so several hours or days, then we see interesting thing. In ALD patient cells, there is an accumulation of very long-chain fatty acids sort of all over the cell, in the cytoplasmic areas, on the membranes. And this is actually a recapitulation of the disease pathology as we know of it on the molecular and cellular level. So basically, we see that very long-chain fatty acids accumulate and potentially incorporate in other membranes, which is probably one of the first steps in the toxicity development. So I would like to just summarize what I showed you today. So I showed you that there are different levels depending on cell types and conditions of ABCD1 and other transporters, which is of course known from the literature, but it was just interesting to me when I started in this field. And then I showed you that we're developing functional probes for ABCD1, and we are trying to improve them for use not only in CRISPR-Cas9 models of ALD, but also in patient cells. And the whole sort of situation here is with hopes of potentially classifying all the variants of ALD. So it will be complementary to what we already know from this amazing analysis by mass spec of different lipid classes. It will be complementary data sets for function of actual protein on the peroxisomal membrane. So thank you very much. This work would not be possible without Stefan Kemp. So I really thank him for introducing me to this amazing community. And thank you to all of you researchers, patients, and carers. You're doing an incredible job. Thank you.